Right. Good morning, everyone. So today I will be presenting the Government of the Canary Islands strategy for the genomic monitoring of biodiversity. So our overall aim is to conserve genetic diversity, which is one of the fundamental pillars of biological diversity, and it is essential for species resilience and adaptability. So therefore, we need to incorporate genetic diversity assessments into long-term monitoring strategies. And so we set up two specific objectives. Firstly, the characterization of biodiversity at the genomic level. And secondly, the integration of biodiversity genomics with policy and practitioner needs. So this is an overview of our strategy where we use, which is based on the use of non-genetic data as well as DNA data to calculate indirect and direct measures of genetic diversity in order to improve the monitoring of biodiversity at multiple scales and also to improve the protection of genetic diversity. So using non-genetic data, we can calculate genetic indicators which offer a way for us to perform rapid and large-scale assessment of biodiversity when molecular data is not available. And then we can also use DNA data to perform more in-depth investigations of species and populations. Both of these provide very different types of information, and both of them are very useful and valuable for conservation management. So I'll start by talking about non-genetic data, which are proxy-based geographic and demographic data, which we can use to derive these genetic indicators that can inform us about the genetic trends, status, and also assess the efficacy of conservation policies on species. So we have established three main genetic indicators so that we can indirectly assess genetic diversity, two of which were developed by the Global Biodiversity Framework. So first of all is the number of populations maintained over time, which is a measure of population extinction. So if we assume populations to be distinct and locally adapted, then if a population becomes extinct, then we can equate this to a loss in genetic diversity. And this refers to a notion that population maintenance is required to ensure the adaptive potential of a species and to prevent species collapse. So following, effective population size is another very well-established indicator that refers to the rate of loss of genetic diversity within populations. So when effective population size, or NE, is below 500, we can expect an increase in genetic diversity loss via genetic drift and an increase in inbreeding. And this will impact the ability of species to adapt via natural selection. However, when NE is equal or above 500, this is usually taken as a threshold for a suitable population size that will maintain its genetic diversity over time. So our last genetic indicator is based on the relationship between genetic diversity and the area of a species. So this is an extension of the species area relationship, which is a foundational concept in ecology. And there is growing empirical and theoretical evidence that shows that we can extrapolate this from species to alleles. And it is a very straightforward way for us to indirectly measure the genetic diversity of species. So for instance, we are routinely using satellite imaging coupled with AI in order to examine species distribution for many species simultaneously. And this allows us to track habitat ranges and their changes over time, which then serve as proxies for population sizes and genetic diversity. So in this example, you can see how we used these technologies to refine our map of distribution of palm trees in La Gomera, where the green dots are the points, are the individuals that we were identified manually, and the red ones were those additionally identified using AI. So this kind of tool provides us with a way to incorporate like more detailed monitoring and to detect very early changes in population sizes that could then result in changes to genetic diversity levels. So briefly, in the Canary Islands, we have a biodiversity inventory that's called Biocan that allows us to track species and habitats, as well as provide the basis for us to calculate these genetic indicators. In particular, Biota provides a lot of information about species, including their distributions. Here you can see an example of the distribution of the palm tree across the archipelago. And notably, each occurrence point is associated with a dated publication that has a reliability or precision associated to that georeference. And this enables us to track species through space and time, which is key for long-term monitoring. So overall, genetic indicators are a very useful way for us to assess genetic diversity indirectly. They allow us to track species over time, as well as report to regional, national, and international policy frameworks, and especially to identify species that may warrant a more in-depth analysis. However, they do have notable limitations, such that they focus only on reduced aspects of genetic health. They rely on assumptions and they're based on thresholds, which is when DNA data becomes very useful for us. Genome-wide studies provide a very comprehensive overview of the genetic diversity of a species, allowing us to examine different parameters such as quantifying genetic load, introgression, population sizes through time. And here we're implementing two approaches based on the use of molecular data, namely the single species and the multi-species approach. So within the single species approach, we are prioritizing the generation of reference genomes for endangered species, and if necessary, couple this with population genomics studies. And some exciting examples that we have at the moment include the start of two projects to generate reference genomes for the Fuerteventura skink and the long-eared bat, as well as a past RAD-seq study on limpets that was key to direct the genetic rescue of this endangered species, and also a mitogenome project on hammerhead sharks to study local adaptation and differentiation. So as part of our single species approach, we're also aiming to barcode all wild species, which includes native and invasive species, with the final aim to build a reference library of barcodes for the Canary Islands that then we can implement in landscape analyses. So another example is that we are currently doing an analysis to identify to the species level 80 putatively invasive marine invertebrates. So moving on to the multi-species approach, our objective is to calculate genetic diversity at different scales. So we want to end up with maps of genetic diversity and phylogenetic diversity, as well as ecological indices derived from molecular data for every island across different taxonomic ranges. And for this, we will be prioritizing a meta-barcoding approach, preferably based on environmental samples, so that we can really systematically track biodiversity over time. So continuing our effort to implement genetic diversity into our decision-making, we are in very early stages of developing a new biodiversity inventory that incorporates this type of data. So we want to facilitate the access of all the molecular data that is available for species, as well as report genetic diversity indices and, if possible, reconstruct phylogenetic relationships between our species. So basically, we could see how molecular data can reveal genetic patterns across taxa, time, and space. So through the single species approach, we can improve our conservation policy, as well as validate our genetic indicators derived from non-genetic data. And through a multi-species approach, we can improve our spatial conservation planning, devise top-down policies that protect genetic diversity, and also identify areas and taxa that are at risk of genetic erosion. So hopefully, these approaches will help decision-making for conservation practice. And lastly, I just wanted to introduce a new initiative to establish a biodiversity genomics consortium for the Canary Islands, where our main aims will be to generate reference genomes for all endangered species, barcodes for all wild species, and also to develop tools for conservation, protection and restoration of species and ecosystems, and finally to promote the sustainable development, innovation and collaboration, particularly at the regional level, where we really want to foster a close collaboration between researchers and the administration. So we hope to be in touch soon about the establishment of the consortium so that we can invite all of the groups working with biodiversity from the islands and support as much as possible. So finally, I'd like to thank the organizers of the conference, everyone at IBNA for their support, our colleagues in the field and at the Servicio, and all of you for your attention. Thank you very much.